All right, today we're diving into a real evolutionary detective story. We're going to trace the history of a single, super important protein back, way back, like 450 million years, all to solve a fascinating biological puzzle. So let's get right into it.
So, what can a fish possibly tell you about your own immune system? I know, it sounds a little strange, right? But the answer, well, it's this incredible story about how life works at the deepest molecular level. It's a story about stability, but also about wild innovation. And the clues? They're hidden inside a protein that's floating around in your blood at this very second. Okay, so every good mystery needs a main character. And for us, that character is a protein called the C1 inhibitor. Think of it as a silent guardian working around the clock inside all of us. So, what does this C1 inhibitor, or C1IN for short, actually do? Well, it's a regulator. And honestly, the best way to think of it is like a bouncer for some of your body's most critical systems. Its main gig is to keep parts of your immune system in check, making sure they don't overreact and cause dangerous swelling. Without this bouncer, things can spiral out of control really fast. It's absolutely crucial. Now, this is where our mystery really kicks off. If you look at mammals, birds, reptiles, basically all land vertebrates, the design of this protein is incredibly consistent. It's like evolution found a blueprint that worked and just said, yep, let's stick with that. But when scientists took a look at the C1 inhibitor in fish, they found something they totally did not expect. It was a completely different story. And that brings us to the first major clue in our investigation, a really stark difference in the architecture of this protein between fish and the rest of us land dwellers. Take a look at this diagram from the research paper. It just lays the puzzle out so perfectly. See those top three lines for mammals, birds, and turtles? They've all got that blue part, the serpent domain, and that little tail. Simple, right? But now look down at the fish structure at the bottom. It's got the same core, but it also has these two extra red segments, IG1 and IG2, basically bolted onto the front. So, what are they, and why on earth are they there? So here's the breakdown. On the left, you've got the version in tetrapods, like us. It's like a simple wrench. It has one main functional part, that's serpent domain, and a little handle. But the fish version on the right, that's more like a Swiss Army knife. It's got the same wrench, but it's also got two extra tools, these immunoglobulin-like domains fused right onto it. And you can really see the difference here. On the left, that's the simple, streamlined design we have. But on the right, wow. That's a 3D model of the platyfish C1 inhibitor, and you can see how those extra domains create this much more complex multi-part machine. So the big question is, why did fish need this upgrade? And that question is what takes us even deeper, down to the level of the genetic code itself. Just when you think you're getting a handle on it, the plot thickens. We have to stop looking at the protein and start looking at the recipe that built it, the DNA. And that's where things get even weirder. I mean, having extra parts is one thing, but how did they even get there? And is that really the only difference between the fish version and ours? To find out, we have to zoom in, way in, on the gene itself. And that's where the story takes another really surprising turn. Okay, this diagram shows the gene structure in different fish. Now, stay with me here. Let's start at the bottom with the zebrafish, which is an older lineage. See that single block labeled IM2? Got it? Okay, now look at the other fish above it, like the platyfish. That one block has been split clean in two. IM2A and IM2B. And what split it? A brand new piece of DNA called an intron that got jammed right in the middle. This isn't a small change, it's a major genetic event called an intron gain. So now we have a whole new mystery on our hands. Not only do some fish have these extra protein domains, but their genes have this new intron, this extra bit of code. So what in the world is going on here? So why would this happen? I mean, why would evolution just shove a new piece of DNA into a gene that's already working? Well, the clue, believe it or not, wasn't found by zooming in even further. It was found by zooming way, way out and looking at the size of the entire fish genome. And get this, here's the evidence. This chart shows a really stunning connection. On the left, you've got the fugu, the pufferfish. It has a teeny tiny genome, super compact. And guess what? It has the new intron. Now look at the zebrafish in the middle. Much larger genome, and it lacks the new intron. The pattern is clear. The fish with the smaller, more squished down genomes are the ones that got this new piece of DNA. So this led scientists to what they call the genome compaction theory. And it probably went something like this. First, as evolution was shrinking down the genomes in these fish, the whole process got a little messy, and it probably caused accidental breaks in the DNA strand. The cell's natural repair kit would then rush in to patch things up. But here's the thing. Sometimes that repair job isn't perfect, and in patching the hole, it accidentally stitched in a new piece of DNA. A mistake, really, but a mistake that ended up sticking around. So, after all this detective work, looking at protein shapes, digging through the genetic code, what's the final verdict? What does all of this tell us about how evolution really plays the game?
You know, the most mind-blowing fact in all of this is just the sheer timescale we're talking about. The core of this C1 inhibitor gene, it's been preserved for 450 million years. That's before the dinosaurs. That's before trees really even took over the planet. Just let that number sink in.
Now, I know this diagram looks incredibly complicated, but the message is actually really simple, and it's so powerful. It's tracking the location of our gene across all these different species, from fish all the way to humans. The best way to think about it is like a street address. So despite all those crazy changes we just saw, the new protein parts, the new introns, the gene itself has stayed in the exact same genetic neighborhood for 450 million years. This is called synteny, and it's just this amazing testament to how evolution holds on to the things that really, really work.
And that right there is the big takeaway. This one single protein tells this beautiful story about two forces in evolution that seem like they should be opposites. On one hand, you have this incredible, profound stability, keeping a critical gene in the same place for almost 500 million years. But at the same time, you have surprising, sometimes accidental innovation that leads to a totally new design. So let's go back to our first question. What can a fish tell us about our immune system? Well, it tells us that the fundamental parts of our biology are ancient, and they're deeply shared across the tree of life. But it also shows us that evolution is never done. It's always tinkering, always customizing, always building on that shared blueprint, leaving behind this amazing trail of clues for us to follow.